Hello students, now we are going to start with the 3 mark question in this video. Already we have completed 1 mark question and 2 mark questions, and already I have uploaded the video. Now I am going to start with 3 mark questions of this CBSE sample question paper. So let's start with the question. And if you are not subscribed my channel, please consider subscribing my channel so that you will get the notification of the all videos which I will be uploading. So start with question number 13. Draw and explain how the nerve cell helps in transmission of impulses. So here the question, we have to draw a nerve cell, that is neuron, and explain how the impulses or the signals will be transferred or transmitted in this nerve cell. So first we will see the explanation of this, and with the explanation we will draw the diagram. So here answer. First point is nerve cells, which are also called as neuron, receive a stimulus at their dendrites. So let me draw the diagram of nerve cell. So this is the diagram of a neuron. So this is one neuron, you can see, and this is the another neuron. And in this neuron, here, this is nucleus. So I'll write it here, nucleus. And these branch structures, you can see, these are dendrites. So I'll write these are dendrites. This structure is called axon, and these are called nerve endings. These are called nerve endings. And this body is called as cell body. This is called cell body. Now this is another neuron. In between these neuron, you can see the gap. So this is the gap between the two neuron, which is very important and which is called as synapse. So this is called as synapse. So now, so according to the first sentence, as I have written here, nerve cells, that is neurons, receive a stimulus at their dendrites. So first these all the nerve cells will receive the stimulus. So imagine this is the stimulus. This stimulus will be received by this dendrites. Dendrites will be the branched one. So once this dendrites receives the stimulus, which starts an electrical signal called nerve impulse. So once it receives that signal, here the electrical impulse will start. That signal will be converted into electrical impulse, electrical signal, and which is called as nerve impulse. This nerve impulse travels through the cell body and down the axon towards the axon terminal. So this nerve impulse which has been caused, this will travel from this cell body to this axon. So this is called as axon. Through axon, this nerve impulse will travel till the axon ending. This whole is axon. This is called axon ending or nerve ending. So it comes till this axon ending like this. Once it reaches this axon ending, or also called as nerve ending, now the next is at the end of axon, the electrical impulse causes the release of chemical messenger. So once it comes into this axon ending or nerve ending, here they prepare a chemical messenger. So here the reaction takes place and chemical messenger is prepared, which is also called as neurotransmitter into the synapse. So this whatever chemical messenger is prepared, this chemical messenger comes into the synapse. Synapse nothing but this is the gap between the two neurons. So this is transmitted to this synapse. Synapse is the gap between the two nerve cells. Once that chemical messenger comes into this synapse, what is the next step? These chemicals cross the synapse and start a new impulse. So these crosses this synapse, this chemical messengers will cross the synapse and comes to again the dendrites of the next neuron. And once it comes to the dendrites of the next neuron, again those all the chemical messengers will convert into electrical signal, nothing but a nerve impulse. That nerve impulse from this dendrites goes to the cell body. again goes to the axon and transfer till the axon ending or the nerve ending like this. Once they reach to the nerve ending, ending, here again chemical messengers will be secreted. These chemical messengers or neurotransmitters will travel this synapse which is present between the existing neuron and the next neuron. Once they travel the graph, goes to the dendrites of the next neuron. And again the process continues. So remember, as I told, once again I am repeating. So first the stimulus will be taken by the dendrites. It will be converted into an electrical signal called as nerve impulse. Those nerve impulse will travel from the dendrites to the cell body, then to the axon until axon ending. Once they reach to the axon ending, here they release some chemical messengers which are called neurotransmitter. These chemical messengers will travel this gap. Gap means synapse. And once they pass this gap, they contact with the dendrites of the next neuron. And here they again convert into an electrical signal. So again prepares a nerve impulse. Those nerve impulse will again travel from the dendrites of the next neuron to the cell body of the next neuron to the axon till the axon ending. And the next again process repeats. And in such a way, any signal stimulus will be transferred from the receptors to the main part of the brain, that is spinal cord or the brain. And by processing there, we will get back the reverse message from the brain or the spinal cord to the muscles or the effectors. So this is the answer for question number 13. Here we have to draw the diagram and explain. So this is the diagram which you have to draw and explain these three sentences which I have given here. Write down these all the sentences in your notes. So now we are going to move to the next question. Question number 14 is, in a genetic experiment, plants with pure round green seeds, pure plant is pure, round and green. So shape is given and the color of the seed is given. So it is pure, so it is denoted by capital R for round and small y for green. Next, were crossed with plant with wrinkled seeds, wrinkled and yellow seeds. So wrinkled is small r and yellow is capital Y, capital Y. You have to be very careful. See how they have given. So here plants are pure, but the first plant is round and green. Round is capital R dominant. Green is small y recessive. Second plant, wrinkled is recessive, small r, but yellow is capital, that is dominant, capital Y, capital Y. Question is, show the gametes formed in the F1, that is self-pollination. First generation we have to show the gamete. And then question 2 is, a total of 144 seeds were produced. Whenever such type of pollination will be done in F2 generation, 144 seeds will be produced, which develops into saplings. Show the ratio in which these traits are independently inherited in these 144 saplings. So means we have to show the ratio of F2 generation, where we can see how these traits are independently inherited. Means here phenotypic ratio we have to show for F2 generation. So just the difference is what the important thing is, what you have to remember, which are dominant and which are recessive, and according to that we have to pollinate it. So we will start with first F1 generation. So first we will do F1 generation. So I'll write it here, F1 generation, and that is nothing but our question number 1. So in F1 generation, what we have to do? We have to take both the plants. So here I am going to write pure round green seeds, and these are denoted as capital R, small y and small y. This you have to write it very carefully. Then the second plant is pure wrinkled, wrinkled, and it is yellow seeds. should not go wrong while writing this. If you go wrong here, then everything will go wrong because F2 generation is connected to F1 generation. If F1 generation goes wrong, F2 will be wrong. So now write down the genes for this. The genes will be capital R, capital is twice, so we will write it only once. Small y is twice, so we will write it only once. Same thing on this side. Small r once and it should be capital Y. See here, as I told you, you have to be very careful. So it is capital Y. Now, these both the plants, we are going to self-pollinate. Now, see, these both the plants are different. So here, whatever pollination is done, it is called as cross-pollination. Cross-pollination. And once we cross-pollinate, what we are going to get? We are going to get capital R, small r, and capital Y, small y. Now, if you see here in shape, there is one capital R. If there is a capital, that is dominant. So here shape will be shown by this capital R, which is nothing but round. So here we can see round shape. If you see the color, there is a capital Y, and capital Y is denoting yellow. So that is yellow color. So whatever seeds we are going to get by this F1 generation saplings, F1 generation seeds, all will be round and yellow color. This we have to remember. Now, question 1 is finished. Now, what we are going to do? We are going to take this F1 generation plant and self-pollinate. And whatever ratio we are going to get, that is called F2 generation. That we have to write it. Answer for the second question. We will see on the next page. So here we are going to start with F2 generation, and I have taken the seeds or the plants which I have got in F1 generation. So we have got round yellow, and same round yellow another plant we are going to take. In that only we are going to self-pollinate it. So that's why I wrote same on both the sides. Capital R small r, capital Y small y. Capital R small y, small r, capital Y small y. Now whatever the genes we are going to get, that we have to write it. While writing the genes, you have to remember the trick like this. Capital R, capital Y and small y. Write it like this, and then you should combine like this. Then we are going to get here, that is capital R, capital Y, capital R, small y. Then you have to combine like this. Then I'm going to get small r, capital Y and small r, small y. So like this we will get the genes. Same thing we are going to write this side also, because in same plant we are going to pollinate now. So I am writing the same genes on this side also. Same gametes here. Now it is ready. Now we are going to do self-pollination. Now see here, there are so many gametes, and it will be very much difficult if I cross it and write it here itself. So we are going to use checkerboard for this. So this is the checkered board. Now write down all the gametes. That is, here I am going to write capital R, capital Y. So this one I wrote. Capital R, small y. This one also I wrote. Then small r, capital Y over. Then small r, small y. Same here also we have to write, because we are going to do it self-pollination. That's why we are writing the same one. So I wrote. Now it is ready. Now go on pollinating, means combine the genes and write down the answer for that. So here I am going to write all. So you can see, if I combine this and this, what I'm going to get here? Capital R, capital R, capital Y, capital Y. So we will write it here, capital R, capital R, capital Y and capital Y. Same, now this we will combine. Now what we are going to do? Capital R, capital R, capital Y, small y. Now this we will combine. Now what we are going to get? Capital R, small r, capital Y, capital Y. Now with this we are going to get capital R, small r, capital Y and small y. This we should not go wrong. You have to be very careful while writing this, because whatever phenotypic ratio we are going to write it, that will be based on this checkerboard only. So whenever we combine, you should be very careful, take your own time and complete this checkerboard. Now same way I am going to start with this one. So write down. So here I have completed the checkerboard. So it will take more time to complete. So that's why I've completed everything. Now we will start writing the physical character of this by seeing the dominant and the recessive gene. Now see the first one. R is capital, Y is capital. So what we can write? Capital R means it is round. And Y capital means it is yellow. So it is round yellow. I hope you're understanding. Here also one R is capital, one Y is capital. So this is also round and then it will be yellow. Same way, this is will be also round and yellow. I hope you are getting what I am doing. So you have to just focus on the capital and the small letter. So see here, this is also round and yellow. This is also round and yellow. You have to just focus which letter is capital. So here R capital round, Y capital yellow. At least one letter there should be capital, so it will be a dominant one. Now see the next. If you focus here, R is capital, so I can write it here round. ROUND. But Y is not capital, Y is small, so it is not yellow, it is green. I hope you understood. Now see the next one. R capital, Y capital. So this is again round and yellow. But see the next one. It is round, but it is yellow. Sorry, it is green. Now again it is R capital, Y capital. So round, yellow. Now if we see down, it is round, but it is not yellow. It is green. Now see the next one. Next one, if you focus, let me take the different color. Now if you see small r, small r. What is small r? Capital R is round and small r means what? It is wrinkled. So I'll write wrinkled. But Y is capital. So it is yellow. This is also wrinkled. But there is a capital Y. So it is yellow. This is also wrinkled. But here Y is capital again. So this is yellow. And if you focus on the last one, it is small r means wrinkled. See, it is wrinkled. But small y means what? It is green. And like this, we have written the physical character by seeing this all the dominant and the recessive genes. Now we have to focus on our that phenotypic ratio. So I'll take the pink color and I'll round whichever are round yellow. So this is round yellow 1, round yellow 2, round yellow 3, round yellow 4, round yellow 5, 6, 7, 8 and 9. So round yellow we have 9. Now next we will see that is round green. So this is round green, this is round green and this is also round green. So I'll write it here round green. And here how many we got? We got 3. So now I will move with the next, that is the wrinkled yellow. So wrinkled yellow, wrinkled yellow and wrinkled yellow. So write down wrinkled yellow. How many we got? We got 3. And the last one, that is wrinkled green. We have only 1. So I'll write wrinkled green is only 1. So what is the ratio? That is phenotypic ratio. Phenotypic ratio means the physical character or the traits are transferred in F2 generation by self-pollinating the saplings from F1 generation. It is in this form or in this ratio, that is 9 is to 3 is to 3 is to 1. So whatever total 144 sapling we are getting in F2 generation, in those all, the traits will be transferred independently in this form of ratio, that is 9 is to 3 is to 3 is to 1. So this is the answer for this question. I hope you understood. Here the important thing is writing these genes and then by writing this checkerboard. So this is the answer for this question. Now we are going to move with the next question. So here we have completed all 3-mark question. Now the next is 4-mark question. For that we will do a separate video. I hope you're understanding all the answers. It will help you to write the answers in your final exam. If you like my videos, share my videos with your friends. Comment the doubts which you have in these all the question papers so that I will solve those all the doubts. I'll prepare the separate videos and I'll upload. And please consider subscribing my channel. Thank you so much.